This is a production of Cornell University.
All right, so today I'm going to talk about my efforts to characterize early blight tolerance within cultivated tomato. Early blight is caused by Alternaria tomatophylla or Alternaria solani. That's a necrotrophic fungal pathogen within the Dithidiomycetes. This disease is widespread. One plant pathologist described it as present wherever tomatoes are grown. Now that may or may not be true, but it certainly is a problem in humid and semi-arid environments. Resistance to this disease is quantitatively inherited, and the mechanisms underlying that resistance remain poorly understood. But that doesn't mean that there isn't substantial resistance within cultivated tomato. In fact, while there is no single gene resistance that provides full resistance, there is quite a bit of resistance that's already been integrated in the cultivated germplasm. And while some mapping studies have mapped QTL associated with tolerance or partial resistance to early blight, they generally have had to do, or they generally have been in crosses with unadapted wild species germplasm, and they haven't really advanced into the breeding programs past that wide cross stage. And so as a result, we don't really have any markers that are predictive for use in everyday breeding. And so that's the goal for this project. That's been my objective for the last year and a half or so, is to develop some PCR markers that are predictive of the major QTL providing resistance to early blight, and then test them in several populations. So what's interesting about the early blight disease of tomato is that the resistance to it seems to be somewhat tissue specific. So we have different responses in the foliage and in the stems. So I'm going to break this down during the course of my discussion today in terms of the foliar and stem tolerance. So starting with this, several years ago, Martha began breeding for Northeast adapted fresh market tomato lines that were resistant to early blight and also resistant to septoria leaf spot and late blight. She integrated in particular a resistance in the stems, but also found that that resistance conferred some foliar tolerance as well under a lower spray regime. But the most marked difference is in the stems themselves. So on the top here, we have the resistant genotype. This is our resistant control. As you can see, there are lesions that are present, but they don't tend to elongate along the length of the stem like they do in the susceptible material. And that's important because if you have if this lesion elongates and hits a leaf axil, the whole leaf tends to die. And in the same case, with the fruits, you actually provide some fruit resistance by having this form of tolerance because the pathogen tends to sort of enter the fruit through the top, through the stem end. You can actually see here in this upper right-hand corner how the fruit is sort of rotting from the stem end out. It's kind of interesting. So in order to map the resistance to this, in order to map that stem resistance in particular, and also to look at the foliar resistance, I worked with 3 parents, and I'm going to be talking about these throughout the course of this discussion. Our first one is 151095-146. That's a Cornell triple resistant line, late blight, early blight, septoria leaf spot. It's the same one that's pictured here. We also have 2 Ohio processing tomato lines, Ohio 7663 and Ohio 7536. They come from David Francis's breeding program at Ohio State. Ohio 7536, for all intents and purposes, is considered our fully susceptible control, and Ohio 7663 actually has some form of foliar tolerance. I should note, though, that neither of them are really truly fully susceptible because they are from a breeding program that's applied and is in a humid environment. So like a lot of breeding material that's meant to become a cultivar, it's never fully susceptible. And so in order to do this mapping population, or in order to do this mapping, I planned out 2 mapping populations in the fall of, or in the summer of 2017. An F2 cross between 151095-146 and 7663, and a triple cross that involves all 3 of these parents. I inoculated them with a backpack sprayer full of Alternaria North Carolina number 10, this is Alternaria tomatophylla. And I did separate phenotyping for stem and foliar traits. The stem is on a 1 to 5 scale, and the foliar is on a 0 to 100 continuous scale. And then I also measured several important, or what I thought would be important covariates, such as fruit size, fruit set, fruit count, and the plant architecture. In general, when you look through the literature, you see that as the fruit size goes up, and as the number of fruit go up, and the internode length goes down, you become more susceptible to early blight. Then from these populations, I genotyped about half of the F2 population and about a third of the triple cross population. I started with GBS markers, but about a year later, it became apparent that we needed some, or we were missing some genotypes in some key areas. And so we threw some SNPs at it using the SolCap SNP set. And so I was able to map resistances to the stem phase of this disease on 1, 4, and 9. The minor effect QTL are somewhere in the range of 6 to 7, 6 to 8%. But the real killer is this guy up here. This chromosome 9 QTL explains about a third of the variation for the stem trait. That's major effect locus. For the foliar form of the disease, we've got hits on 1, 5, 8, and 9. They're all about between 5 and 12%. They're on the lower end, but that's consistent with the literature and consistent with what we see in terms of the distribution for this phenotype. There are also significant interactions between the QTL and between these QTL and other regions in the genome, but for the purposes of keeping this discussion short and concise, I'm only going to be focusing on 3 QTL. That's on 1, 5, and 9. These are our biggest effect additive loci. And I want you to note also that the QTL on chromosome 1 and 9 also pop up for this stem phase of the disease. So to confirm the QTL effects, we did a few things. To start with, I looked at the genotypes for the F2 individuals that were used for mapping this trait, and I selected, based on these QTL regions, for F2 individuals that were homozygous for all 3 of the QTL regions in all possible combinations where possible. So a 1 in this case is the CU071226 allele, or sorry, CU151095-146 allele, and the 3 is the Ohio 7663 allele. And I'm going to be using this coding throughout as well. And I planted the F23 families, so the progeny of these selected F2 individuals, in a replicated trial this year in Terwilliger, and I sprayed them again with Alternaria entomatophila, and we did mapping, or we did phenotyping for the stem and foliar traits. And to start with, if we just look at the distribution of the stem phenotype about the marker scores, and so the genotypes here again are 1, 2, and 3, we see that there is a significant difference for the phenotype between the markers. And actually, when we did that additional genotyping using the SolCap SNP set, the locations for the QTL shifted a little bit. And so here are the updated QTL locations. I told you that I did select for homozygosity at the QTL, but unfortunately, when we changed those positions, we introduced some heterozygosity at the QTL locations. But actually, the fit on chromosome 1 is significantly better. But what may be more convincing is if we just rank the F23 families by their phenotype. So in this case, we've got the stem infection. We've got families, F23 families on rows. And as you move down the table, you have increasing disease in the stems. The Tukey multiple comparison is used to differentiate the families statistically. And this coding I've used here is a single plus indicates homozygous for the resistant form of the allele, a plus minus indicates heterozygous, and a minus indicates homozygous for the susceptible allele. And these are the 3 loci, or QTL on chromosomes 1, 5, and 9. And importantly, what we see is on chromosome 9, that major effect locus is perfectly associated with the trait. So that's definitely encouraging. We did the same thing for foliar, ranking again from most resistant to most susceptible. And you again see that the distribution of resistant alleles tends to be associated with plants that are resistant, in fact. And again, for the markers, chromosomes 1, 5, and 9 QTL, we do see significant differences on the phenotype as a function of the marker scores. Looking at this a different way and using a different population, I generated, I generated a backcross 1 individual. And so this is back cross to the recurrent parent, and we use background genome selection to clean up this material as much as possible. So actually in selecting, we were able to eliminate all but 3 chromosomes from play, essentially. So the remainder of the genome is fixed homozygous for this Cornell recurrent parent genome. And of those 3 chromosomes that are still segregating, 2 of them are chromosome 5 and chromosome 9, which are where our QTL lie, or 2 of our QTL lie. So we put a segregating population out, which is the self-progeny of that BC1 individual. And for the stem trait, again, we see nice separation here, where as you move from the Cornell genotype to the Ohio genotype, you become more susceptible to the disease. And again, for the foliar, we looked at the chromosome 5 and chromosome 9 QTL. Again, statistically significant separation. And then finally, for the last thing that hasn't been yet confirmed, this chromosome 1 locus, we chose an F2 population, or excuse me, an F2 individual from this cross, this Ohio 7663 by 1095-146, and crossed it back to a different Ohio parent. This F2 happened to be fixed for chromosomes 5 and 9, and so we're actually amplifying artificially the effect of the chromosome 1 locus. And we do see a significant effect there. So I hope that I've conveyed that there's statistically significant results for these 3 QTL. But what about visual or practical significance? Because we want to use this in a breeding program, and if we're going to say a line is resistant, if a farmer or a grower receives this material that claims to be resistant and it's not, we're in trouble. So for that, I'm going to take you on a virtual journey back to Freeville, New York in the summer of 2018 on a sunny day in August. And we're going to walk through the replicated trial in an abbreviated form. So if we start with a plot, an F23 family that has 0 of the QTL, it's fully susceptible. As you can see, the plants are not happy. There's no foliage left. The stems are all infected. It's dead, total loss. We add in a chromosome 1 QTL. We get some retainment of foliage, but in general, the plants still look pretty awful. Same thing with the chromosome 5 locus, or chromosome 5 QTL on its own. If we add in the chromosome 9 QTL on its own, again, this looks pretty awful, but if we zoom in on the stems are happy, they're green. So as expected, it provided a lot of stem tolerance, but not a whole lot of foliar tolerance. If we start adding QTL together, though, then we start getting measurable results, or practically different results. We see an enhancement in the ability to keep the foliage. But in this case, we don't have that chromosome 9 locus, and so the stems are still dirty, still infected. If we add in the chromosome 5 and chromosome 9 together, you get about the same level of foliar tolerance, but you can actually see there are fewer of the fruit that are rotting prematurely on the black plastic here. You can see that in terms of these orange and red fruits. They tend to actually, at this early stage in the season, if you see that, it's usually because the infection has actually separated the fruit from the plant, and so it's ripening on its own. And when you add all 3 of the QTL together, you get a plant that looks pretty darn good compared to that initial susceptible plant that was completely dead. And so, and also I want to point out that I inoculated this with a backpack spray full of material of the pathogen. I sprayed it with overhead irrigation, and I to basically enhance the disease. And I didn't provide any chemical control whatsoever, because I wanted this disease to spread like wildfire. So if this plant is doing this well, or if this plot is doing this well, given those conditions, you can imagine that in a low to moderate spray regime in a farmer's field, it could actually be doing pretty well. And so with that, just talk briefly about next steps for this project. So I've looked at the effects of the QTL, but I haven't totally controlled for all the background effects yet. And so for that, we really need to generate some neoisogenic lines that are in the background, ideally of both the Ohio and Cornell material, and test the QTL effects on their own. And so we're in the process of doing that. I'm also going to be doing some selective phenotyping. So I'm going to plant out 1,000 or more individuals. We're going to use genotyping to identify some individuals that have useful recombinants. Then I'm going to phenotype using a greenhouse screen you actually see up here that I've got running. And we also received some sequence data from the 2 parents. And so I'm going to be going through and looking at the sequences in the QTL locations to try and identify some candidate genes. And I'll be adding in some digital phenotyping work to try and enhance this. I've actually begun using this tool called the Phenom from Mike Gore's lab. And hopefully I'll get to analyzing that in the next couple of weeks. And then finally, we're going to be creating products with this. We want to be able to use these markers to generate material that's resistant to early blight, late blight septoria, and bacterial speck and bacterial spot. The bacterial side of the project is another part of the breeding that I'll be talking about next year. And so with that, I'd like to thank my lab. I'd like to thank David Francis's breeding program for providing some of the material and some support, and my graduate committee. And I'd like to thank my funding sources, including Michael Mazurik, who let me TA for his class in the spring of the spring of this year. All right, thank you very much. Are there any questions? Yeah.
I got a couple. One, there was the backcross population. You seem to lose a lot of signal. Do you give any explanation for why that was?
Yeah. So remember I told you I planted out a triple cross population? So that was supposed to be a BC1 back to the Ohio parent, and that's what this is supposed to be. It turns out the C dot was not what we thought it was, and it was actually an F2 selection. And so a lot of the genome is already fixed. Okay. So that's why I don't show an effect plot here, because it's artificially escalated, because a lot of the genome is fixed, so we're not accounting for that.
And then I guess the second one was, it was nice to see all of the stacked genes and the effects that they have. Is it completely additive? Or do you see that there's epistasis between those loci?
I do see epistasis between the loci.
Cool.
Yeah.
Any thoughts?
Well, I have a few thoughts, and I have some concerns as well. So as I said earlier, the early blight trait seems to be associated with several plant architecture traits. And so I want to be sure that what I'm mapping is not just a change in internode length. And so if it's a change in internode length, you can imagine that there would be an interaction potentially. Like if it was on chromosome 1, you would expect to see an interaction between the internode length and chromosome 9. Stem tolerance QTL, and in fact you do. And there are actually pairwise interactions between all 3 of the QTL. So I have begun digging into that, but I haven't fully fleshed it out yet. So I really need immortal populations or nils to test this properly, which is what I'm doing now. Yeah, Kevin.
So with your plots where you had all 3 of the QTL stacked up,
Uh-huh.
The plants did look great, but they also looked like the fruit was later in ripening.
Yeah.
What's up with that?
So that is another association that you see with early blight resistance. And I'm not sure if this is due to the early blight resistance or due to the fact that the fruit aren't separating from the plant. So as the infection sort of gets through the peduncle, it separates the fruit, and then the fruit ripens prematurely. And so in the super susceptible material, you'll see a lot of ripe fruit, right? But it's not actually ripe. In general, the seeds wouldn't be viable. And when you touch it, the fruit's it's not actually there, right? It's not physiologically ripe. It's just red. So again, in order to test this properly, we need immortal populations, because I need to be able to plant this out in a inoculated and uninoculated trial and look for differences. It's in the future. It's on the horizon. Yeah, it's here.
Know about the population genetics of the pathogen much? I mean, like obviously it doesn't have races, it doesn't sexually reproduce in the wild. Are there like isolines or like common lineages that dominate regions? Or
My limited understanding of that is that it does not frequently, if at all, reproduce sexually in the wild. And I can't answer your question about lineages necessarily. There are two species of this, Alternarius monophylla and Alternarius solani. 10 years ago, they were the same species, but they've now been separated based on, I guess, somewhat host specificity. Whereas solani is primarily a potato host and monophylla is primarily a tomato host. Any other questions? Oh, I guess we're out of time. Never mind.
Sorry. Thank you for that presentation as well. I wanted to let you know, guys, yes.
This has been a production of Cornell University, on the web at cornell.edu.